Okay, I hope this doesn't turn off. Let's go over these notes as quickly as I can. The most important ones are stop, and I go over them. The not so important ones will flip over. Okay, this is something you probably learned in your, I don't know, high school or other classes. This is sperm. When sperm enters the egg, of course, leave everything behind. The mitochondria are left behind, and just the nucleus for the tail of the sperm stays behind. Just the nucleus of the sperm enters the egg. The name of the process is called fertilization. That fertilized egg, after one sperm enters the egg, that the name of the egg is called zygote. The fertilized egg is called zygote. Now zygote has complete set of chromosomes, 23. Again, we're talking about human, different animals, different number of chromosomes. In case of human, 23 chromosomes from sperm and 23 chromosomes from the egg, 46 total. So you have 46 chromosomes, all from your head to your toe. You have 46 chromosomes, except right now the male who are making sperm, after they made, the sperm goes to seminiferous tubules, right? Then he has 23 chromosomes. The female, after they make the egg, there is another meiosis right before this enters another, but anyhow, 23 chromosomes. I don't want to go to details. Okay, reproduction, main event, gamete. Gamete is sperm and egg. It's called a gamete as a generic name. Gamete is a generic name. It means sperm, egg, pollen. Soon you're going to deal with it. Pollen, not much now, but soon. Okay. Fertilization is the process of sperm coming to the egg. It's called fertilization. Cleavage, it is when you have an egg, you have one cell, fertilized egg. Somebody took the two cells. By the way, I guess they want to hang it from their bumper in the back of their car. You've seen those? They hang things from the bumper. I guess that's what they took, the two cells. But after one cells, two cells, and four cells. Is that right? I don't have the two cells. So the four cells, it is, the cleavage is occurring. The cells are dividing. That's mitosis. It's a cleavage. The process of cell division in embryonic stages is called cleavage. Then gastrulation, when you have you have a ball of cells like this one. You have morula. Remember, one zygote, and then 2 cells, 4 cells, 8 cells. Is that right? After 8 cells, 16 cells, morula. Okay, and then after morula, you have a few stages of morula. Again, then you have a ball of cells, which inside of it is fluid-filled, hollow. Then this is called a blastula. In animals, in humans, it's called blastocyst. Okay, but these cells, this is morula, a ball of cells that it is full of cells. It's not hollow. Inside of it is not hollow. And if you look at it, morula, same size as egg. Morula, same size as blastula. Same size, but the cells are smaller. I hope you can see that. And then after blastula, you have gastrula. When the cells invaginate, the cells are dividing and dividing and dividing. The cells are dividing, and then they fold inward. Still dividing. I'm not talking about cleavage furrow. Cleavage furrow is something else you learn in Bio 1. Yes, but I'm not talking about it now. So when that invagination occurs, as soon as the invagination of the cells occurred, then that is called gastrula, which is right here, or gastrulation is the name of the process. This is gastrula. You have invagination of the cell. This portion right here is called blastopore. This is called blastocele. And then this keeps going, and this is still gastrula. Keeps going and going and going and going and going until the other end. Then you have a tube. That tube is intestine. I hope I'm making some sense. We went over that. Okay, gastrulation. Neurulation. What do you think neurulation is? Formation of your neurons and nervous system, brain, spinal cord. Okay, and then organogenesis. Formation of what? Genesis, it means formation of what? Organs. Okay, And then finally growth, which is, that would be the embryo, and this would be what? Fetus, is that right? We talked about that. I did talk about it a little bit this morning. Embryology, study of embryo, fertilization, again, zygote. Parthenogenesis, it means some animals do that. Salamander, frogs, some salamanders, some frogs, some fishes. The one that we're studying here is rotifer. One animal we are going to study in here, rotifer, they undergo parthenogenesis. It means sperm is not needed. The egg itself going to multiply and become an adult organism without sperm. The name of the process is called parthenogenesis. Okay, I hope I'm making some sense. No sperm involved, just the egg itself, without the sperm, without fertilization, becomes a salamander, becomes a fish, becomes a frog. Some species, not all of the frogs, not all of them. Okay, example, rotifer, crustaceans, some crustaceans, insects, some insects, fishes, some fishes, lizards, etc. Okay, what is cleavage? Cleavage is the process of nucleus and cell division in zygote, mitosis, after zygote, pretty much. And then types of cleavage, there are 4 main ones: radial, spiral, discoidal, and rotation. Those are the 4 main different types. There are 2 more, so it makes it 6, and I will go over that. Let's go over it. Spiral cleavage. The definition of spiral cleavage is, you should know. That's the definition that you should know. The first 2 planes of cleavage are vertical and produce blastomeres of equal size. The third cleavage is horizontal and makes blastomere are unequal. So what am I saying in here? If you look at my diagram, and this is spiral. What does it say? Spiral cleavage, the first 2 planes, so you have a zygote, right, divides, you have 2 first plane, the first planes of cleavage are vertical, sorry. Well, it depends which, I want to draw it just like, here are vertical. You see what I'm saying? It's a vertical. And then, what is the third plane? Blastomeres of equal size, the third cleavage horizontal, and so you have a horizontal, this one, I'm dividing that one horizontal, and then of not equal size, I'm trying to. So that is spiral cleavage. And then of course they're showing to you right here, not a good diagram, I don't enjoy these diagrams because they're not showing it to you the way I want. Then radial cleavage, radial on the other hand, the 2 main ones that we are going to talk about it for most of semester. Okay, the first 2 cleavage, you have zygote again. Then you have the first 2 cleavage are horizontal, so it would be like this. I hope I'm making some sense. And then and produce equal blastomere. The third plane of cleavage is also horizontal. So you have something like that. It's all horizontal. Okay, so that's the best diagram I can draw and correlate it with what you have. Then it would be something like this. They're showing it to you. Oh, one other terminology maybe I should mention is holoblastic versus meroblastic. Meroblastic, look, of the holoblastic, there's not much yolk in there, so the cleavage is complete. I hope that's what they call it, holoblastic. Meroblastic, you have a whole bunch of yolk, a thick amount of yolk. This is your zygote. This is the first cleavage. A cleavage occurs only partially. It does not go all the way to the end. That's why they call this a meroblastic type of cleavage. So there are 2 types. Some animals have a lot of yolk, like birds, and some animals do not have much yolk. The egg does not have much yolk. They undergo holoblastic. So you do have, like this one, radial holoblastic. It is possible. Okay. So But again, for lab practical purposes, for here, I don't expect you to know like these models. Where is the frog? Frog model right here is a meroblastic. It's not a complete. They don't show it very well in here. But the starfish is holoblastic. Don't worry about it. Rotational is the case of human. After the second cleavage, one pair of blastomere comes in a right angle. As they showed it to you, to your classmates talk about it. It means they come in a right angle. This is, it is in an angle. I hope I'm drawing it a little bit out of wacko for you guys to know what I'm talking about. So it's an angle. It's not horizontal or vertical. It's none of these, and it's that in an angle. That's pretty much. Again, how do you know, Amir? Everything is round. How do you know something is in an angle, it's in a horizontal, it's vertical? It's by the vegetative pole and animal pole. But again, I'm not talking about it here. That goes a little bit more detailed if I have to talk about that. These yolks they have, like here, this frog model, if you look at the bottom, that's a big yolk. So that would be the vegetative or animal pole, vegetative pole, animal pole. So based on those, they determine this is horizontal, this is vertical, this is discoidal, so on and so forth. The other two are easy. I will go over that. So here's a right angle. They're showing it to you, and of course that's discoidal. The cleavage is restricted to small discs at one end of the embryo. You don't have to worry about vegetative pole and animal pole on these two. The next two on discoidal to determine its horizontal or vertical. No big deal there. The cleavage is restricted to small disc at one of the embryo, like birds. They have a whole bunch of yolk. What do you guys think? This is meroblastic or holoblastic? Yeah, it's a meroblastic. It's a meroblastic. The cleavage is not complete. Birds have so much yolk that it is a meroblastic type of. The other type of cleavage is superficial. Yolk is in the center, and cleavage is in the outer surface. An example of cleavage in invertebrates, radial or regulative spiral, protostome, mouth first. I think I talked about that. Protostome animals, quickly, since you guys want to know if this is the blastula. Okay, not blastula, gastrula. This is the gastrula. You have invagination of the cells here. If this becomes the mouth, then that is a protostome animal. The other end, eventually this becomes this. Am I making sense a little bit? In case of human, it takes 4 or 5 days, as you mentioned it. I'm not worried about those dates. You don't have to memorize those time zones, timelines, how many hours, how many days, morula, blastula, gastrula. Don't worry about it. Just know the definition. Okay, so at the end, when it comes, the tube comes together, this opening right here is the mouth. That's a protostome animal. If this opening is anus, that's a deuterostome animal. This is the model of starfish. I labeled them for you guys. So starfish is deuterostome animal. So this becomes anus. This is the model of deuterostome animal. The other model we have here is a frog. Frog is also deuterostome animal. Okay, so protostome, as you know, deuterostome, mouth, second. 4 types of yolk. If I run out of question, I threw one question in there. We have isolethithal, starfish and human, same amount of yolk as the vegetative cells and so on and so forth. Mesolethithal, moderate amount of yolk. Telolethithal, like birds, they have a lot of it. Centrolethithal, it does not say anything about how much yolk has it. It says the yolk is in the center of the cleavage, and blastula is a hollow mass of cells. Okay, blastocele is the space right here. This is blastocele, and some of you confuse this. They say, "Oh, Amir, this is a blastocele. This is also blastocele. This is blastula. This is gastrula." Am I making some sense? Everybody can see this. Sarah Beth, can you see this? both the hollow space inside fluid field is called blastocele. Some of you think this is gastrula, so this must be called gastrocele. No, I should not even say it. The wrong words when I say it, somewhere in your brain is registered. I should not even say that. But anyway, I made a mistake. Okay, so what is gastrula? Having 2 layers of cells within an opening. Yes, that is true. Invagination, archentron, coelomic vesicles. Now gastrula is an embryo and has 3 germ layers: ectoderm, endoderm, mesoderm. Diploblastic, triploblastic animals. Know the fate of Blastopore and Chromosome and Germ animals. Okay, what am I saying in that PowerPoint? On everything in there that you should know. Again, referring to, where is my other model? Did I lose it? Oh, not there. Okay, so what am I saying? When you have, do you see any other color in here? Magic. I want to show you magic. You guys like magic? Do you see any other color? Ida, do you see any other color? No. I'm learning your names. Jessica. No other colors. But have they gone wacko in Waco? Why they come? They have 2 colors in there. Different germ layers. What does that mean? It means the yellow ones develop to become something else. And what is that something else in these models? Most of these models, yellow is indicative of endoderm. The brown is indicative of ectoderm. And I will tell you what they are in a minute. And then if you see red or orange or pink, I told you I was a good receiver, is mesoderm. Anyway, if you guys want to play football, I'm ready. 2-hand touch. Or flag football. No tackle. My days of playing tackle football is over. And of course, I'll play receiver. No other position. Not even a quarterback. Eric, you can say quarterback. Okay? Right here is red, orange, pink is mesoderm. Okay, so what are they? What am I saying in here? Invagination occurs. Is that right? Invagination is occurring. And then it goes from one end to the other end. The yellow becomes the embryonic gut, intestine. And we have a term for that. The embryonic gut or intestine is called archentron. Archentron on your quiz on Thursday means embryonic gut, intestine. Okay. And then coelomic vesicles, you have them, and I will show it to you later on. They are formed on the sides. You don't have it on these models. You have it on the sides. It's called coelomic pouch or coelomic vesicles. Now gastrula is an embryo and it has 3 germ layers. Of course, in this model, where is it? In these models, gastrula only has 2 germ layers, ectoderm and endoderm. Ectoderm and endoderm. You do not see mesoderm on these models. But on this model, right here, this is a frog model. On the frog model, you do have all 3 germ layers, and this is gastrula. This is gastrula. You have the red is mesoderm, all of the yellow is endoderm, and the brown is ectoderm. Okay? And of course, the pink is notochord, which is mesoderm. And I'll show it to you. Pink is mesoderm. It came from mesoderm. Okay, so on this model, this is the frog model. Maybe quickly I should go over it. You have, because I'm going to refer to it now. Now you have the zygote, and then you have 2-cell stage, and then you have morula, and as soon as you see the opening, you see that hollow space inside. You guys see that? That's blastocele. This is blastula, not morula anymore. They cut the ball. They cut the tennis ball right in the middle to show you that you from outside I cannot see that inside is hollow, right? On the slides you can. On the slides they didn't cut it for you, but when you look at the starfish composite slides, you should determine which one is gastrula, which one is blastula, which one is morula. Okay, so from here I see the space on this model, so this is a blastula. And after blastula, again, this is another blastula, empty space inside, blastula, empty space inside, This is out of order. This is again, you don't know it's blastula, but they just want to show you to you. They cut it in half for you to show you what is blastula, what is. There's still blastula or gastrula, God knows what that is, unless I open inside of it, and voila. This is gastrula now. This is different than these guys. Am I making some sense? This is blastula. This is a few days later. This is gastrula. Remember, it is in that order. Zygote, morula, timeline order. Zygote, morula, blastula, gastrula. Embryo, fetus. Oh, neurula. We have a neurula as well. I'll show it to you in a minute. So you have here, you have the yellow is embryonic gut, notochord. You have the mesoderm, yolk plug here. The white portion here is called yolk plug. After gastrula, you have neurula. Neurula, you have a neural plate. Look at this one. This is, if I show it to you in a lab practical exam, this is a neurula. Neurula. Okay? And then they cut it in half for you. So this is neural plate or neural crest. You do have notochord on the top, which came from mesoderm. Question number 8 in last lab practical exam. The mesoderm, endoderm, and everything brown in here is ectoderm. Your nervous system, neural plate, your nervous system comes from where? From ectoderm, endoderm, mesoderm. Oh, right. Nervous system comes from ectoderm. Your brain, spinal cord. Okay? So now you're beginning to have some idea what is an ectoderm, what is endoderm, what is mesoderm. Ectoderm outside. Ectoderm brown is outside. Endoderm more likely inside, and mesoderm in between. Am I making some sense? Meso, middle, your prefix suffixes. I don't know, you studied it or not. You saw it in last exam, you had 6-7 points. Yes, Patrick, please.
So the neurula, it would be obvious from the outside to see since it has the
right, neural plate.
Would you be able to see for the gastrula the invagination from the outside? Or
Yes.
is it
Yes, you always see gastrula, the invagination, or in this case, in case of frog, you do not have it, you have a yolk plug. They call it yolk plug. You'll be able to see it from outside by seeing the yolk plug. We do not have slide of frog, but we have slide of starfish. And then of course we have slide of chick, 24 hours chick, 36, 48, and we have the hours. Okay, so let's move on and then know the fate of blastopore. That's what I mean. The blastopore, right here, the blastopore, does it become mouth or does it become anus? If it becomes mouth, then it is a protostome animal. If it becomes anus, it's a deuterostome animal. I hope I'm making some sense. Alex, am I making some sense? Okay, diploblastic animal, yes. Well, you had a question?
No, it's clear as mud.
Okay, clear as mud. Diploblastic animals, they are animals like starfish, that they have ectoderm and endoderm only. Those are primitive animals in evolution, animals that evolved first. So the first animals that evolved, and we're going to watch some videos on them, they are diploblastic animals. Hydra is an example of it. Hydra is a diploblastic animals. When animals evolved more and more and more and more, then they became triploblastic animals. They had 3 germ layers: ectoderm, endoderm, mesoderm. Okay, don't think this model, on this model, starfish has 3 germ layers. Don't think this is a diploblastic animal. No, this is a triploblastic animal. This is a model of starfish, and still it's a triploblastic animal. Okay, then I will go ahead and give you a more detail of this one, and then you should know that. Main stages of morula, okay, 4 days. Again, don't memorize the days. You want to, that's fine. Blastocyst, 5 to 7 days. Gastrula, 8 to 10 days. Embryo, I guess I do not have Neurula stage in here, 8 weeks. And then after 9 weeks, it is a fetus. And I gave you this morning a little bit of difference between what is an embryo and fetus. So you can look it up online and share it with us next time. That would be a sign of a student who is really into it. You're passive, it's okay. We can be, I don't know. I said no. The differences between protostome animals and durostome animals. Lophotrochozoan is a group, is a clade of animals. Later on, I guess we will talk about it. But these are the main differences that you mentioned. Spiral cleavage are in protostome animals and durostome animals, or radial cleavage, and so on and so forth. I will talk about what is schizocilus and intracilus here. Not a good slide. Here again, protostome animals. This slide is showing you that is schizocilus. Schizo, it means split. And intracilus, it means by formation of intestine, intracilus, and I will go, none of these slides are great for showing you the difference between schizocilus and intracilus. But before I get to the next slide, I guess, which is a good one, that's my favorite one. Okay, so this is, imagine, this is a cross section of the animal. You take an animal and you cut it through here. Am I making some sense? A cross section, and you're looking from top. That's what these are. Am I making some sense a little bit? How you're looking at these animals. You're looking at the animal, cutting it right through the gut, and then you're looking from top. Okay, let's go. This is the best one. So forget this one for a few seconds. Let's talk about the top one, and then I'll come back to the bottom one. The schizocilus animals, which is an example of human, we human are schizocilus, if you can relate more. What happens, this is, do you remember, this is the gastrula stage. You remember? Blastopore, archentron, blastocele. Do you remember all of that? Then, when it goes on and on and on, right before, after the gastrula stage, before neurula stage, for example, the cells right here, some cells develop, early cells, they start developing, and that's what they call it a schizocilus. Schizo, it means split. The cells split, but not in an organized fashion. So they're splitting, the cells are splitting, and then later on, they form vesicles, if you would. They call it coelomic pouch. Inside of it is coelom, and outside of it, the red is indication of what germ layer? Mesoderm. So coelom, by definition, coelom, developing coelom. Coelom, by definition, is the space between gut and ectoderm, skin, gut and epidermis, which is completely surrounded by mesoderm. So the question is, where did the coelom came from? Where the coelom came from? Did coelom came from splitting cells? In some animals, schizocilus animals like us, or it came from outpocketing. You see the same thing as upstairs, right? Downstairs, upstairs, same thing. Intracilus animals are same as these. You have the blastopore, archentron, blastocele, ectoderm, but look at the red. The red is formed by outpocketing of the cells of the gut. They're not randomly split. They form an out pocket, okay? And then eventually they separate from gut, and then you have end product is same thing. That's why we have intracilus animals and schizocilus animals. Intracilus animals, you have starfish. Starfish is an intracilus animal. It's a durostome animal. Generally, this is again just like your heart. You have pulmonary artery, pulmonary vein. There are exceptions. You remember that? Generally, animals that are Protostome, they are schizocilous. Protostome animals, they are schizocilous. We are an exception to the rule. We are deuterostome, but we are schizocilous. Animals that are deuterostome, like starfish, they are deuterostome, they are enterocilous. That's a general rule. Animals, if you look at, that's what I'm talking about. Protostome animals, you remember that? Mouth came first. What do you think they are? Schizocilous. That's a general rule. There are exceptions. Like your heart, there was an exception, pulmonary artery, pulmonary vein. Animals that are deuterostome, they are enterocilous. Good example of that. Good example of that is starfish. Example of this one, earthworm, protostome, arthropods. They're all schizocilous. We human are like this. Of course, we human have to be always. You all know what I'm talking about. I want to make some sense. We are deuterostome animals. This was formed second, right? In your mother's womb, this was formed second. But we are schizocilous. Exception. Anyhow, I made some sense. I hope all of these PowerPoints are making. You plowed through them last night, this weekend. You were wondering, what the heck is this? What is he talking about here? What these PowerPoints mean? Schizocilous, enterocilous, I cannot even say it. Hopefully after today, everything has been ironed for you. You know, you all do ironing? Jose, you don't do that? Wait until you get married. You do all of the ironing, and cooking, and doing the dishes, and raising kids. What else is that? Cleaning the house, did they say that? Cleaning outside, doing taxes, change oil. Did they mention everything? There's still some left. Take out the trash. That's a given.
Build a house.
I'm not saying you. Huh?
Build the house.
The house?
Yeah, have to build it.
Yeah, build the house.
Yeah,
yes.
have to.
From scratch. Build the house. Okay, so I hope I'm ironing things for you if you did everything before you come in here. I hope. Sarah, am I making some sense? I hope. Okay, this is the fate. This is you should know. Don't say, oh, this is not in the book. He's not gonna ask that. Everything that I pull out of my sleeves, is that right? I make it for you. You know it's gonna be the exam because I'll work hard to put it together. Okay, so you have blastula becomes gastrula. Gastrula, then you have 3 germ layers: ectoderm, mesoderm, endoderm. Ectoderm, endoderm, mesoderm. You all know what I'm talking about. It's written in here. Then you do have ectoderm develops to epidermis and associated structures like hair, nails, and also ectoderm becomes your brain and nervous system. This is a very, believe me, whenever you take embryology, when you go to Davis or UOP or any other institution, this is the watered down. They talk about this for the entire semester. This is the what, but you should know this. So, and then of course, mesoderm becomes notochord, mesenchyme, which are loose microorganism cells, become your dermis. The dermis, you all know what is the difference between dermis and epidermis. Is that right, Ariana? You learned that in first exam material. I'm not saying bio 1 anymore. I'm saying first exam. You were with us or you were not with us, I don't know. Okay, but anyhow, mesoderm, bones and cartilage, of course, they come from mesoderm. And circulatory system, your heart, blood vessels, they come from mesoderm. Somites, you do have it on the chick slides. You have outer covering of the internal organs, muscles, excretory system. What is excretory system? Anybody? Yeah, the kidney. Your urinary system. Now, from now on, we start saying excretory system. So it fits, and Patrick mentioned last time, but I'm going to go over it a little bit more here soon. Not this exam, but next exam. What is excretory system? Endoderm, inner lining of GI tract. GI stands for what? Gastrointestinal. Gastro is stomach, intestinal, small intestine, large intestine. Glands, you studied them. Thyroid gland, pancreas, liver, so on and so forth. Inner lining of respiratory system. You studied all of them in first exam. Everything in first exam. You are familiar with everything in there. All you have to do, put it together, which comes from ectoderm, endoderm, mesoderm. There are 3 germ layers. Not the germ like bacteria, viruses. We're not talking about that germ layers. We're talking about germ layers that they are embryonic cells. Okay. Okay, this is, I gave it to you in the last one. If I run out of questions, I will ask you. But you do have chick slides, which is prosencephalon becomes telencephalon and diencephalon. Mesencephalon, cephalon, it means brain, becomes mesencephalon, and mesencephalon becomes midbrain. Oh, I forgot. Telencephalon becomes basal ganglion, olfactory, which is smell, and cerebrum. All of this big portion, the biggest portion of your brain is cerebrum. Remember we talked about that? Has gray matter, white matter. And then pineal gland, we didn't talk about it, very important in biological clock. Diencephalon, thalamus. We have hypothalamus and thalamus. And then hypothalamus, of course, midbrain. And then you have a rhombencephalon. Rhombencephalon becomes metencephalon and myelencephalon. Myelin, it gives you impression of what? Myelin sheath, you remember that? A lot of phospholipid in here. Then you have your metencephalon becomes cerebellum. You remember the cow dung? We talked about it back here. And then pons. Now you know why I asked you to know that stuff. Pons, cerebellum, medulla oblongata. First exam material, you remember that? Okay, most of it. I didn't ask you to know hypothalamus, but it's given. Everybody knows about hypothalamus and thalamus, and they're right inside of the brain. And pineal gland, we didn't look at it, but olfactory, most of it either you know from your past life or we already looked at. Amniotes and amniotic eggs, reptiles, birds, and mammals, they have amniot, chorioallantoic membrane, amnion, allantoius, and chorion. Right here, what is happening with